Thank you. It's my pleasure to participate in this. It's been a wonderful society. I came to my first meeting in 1986 and immediately felt that this really is the society that's most appropriate home society for the field of DNA repair and genomic maintenance. Basic research in these fields provides mechanistic underpinning for the genetic toxicology and mutagenesis testing groups. The annual meeting provides a forum for communication between students and specialists in all of the broad areas impacted by DNA damage and repair, not just the clinical ones, as with AACR meetings, which are also enormous, and we have a comfortable working size group for a meeting. It could be larger. I think 1,000 would be good, but it's a very good opportunity to meet people and discuss. Information from genetic toxicology and environmental concerns prompts researchers to target relevant areas for detailed study. And the EMGS facilitates communication of new discoveries and methodologies in the area of genomic maintenance to researchers in related fields and eventually to the public. On the chemical stability of DNA, Jim Watson in 1965 wrote in his book, "Once atoms are incorporated into DNA, they do not leave it as long as healthy cell growth is maintained." Well, this was just under the radar because the year before, Dixetlow and Carrier had shown loss of finding dimers from DNA in UVA-irradiated wild-type bacteria, but not in Ruth Hill's mutant. So that was really the beginning of excision repair. But 22 pages later in the same issue of PNAS, Boyce and Howard Flanders reported the same kind of result with K12 E. coli strains, in which they showed lack of dimer excision in wild type, but in a number of UV-sensitive mutants, that they were deficient in removing dimers from the DNA. Now, of course, this step is not repairing the DNA. This is generating another lesion, a gap in the DNA. And David Pettijohn, my first graduate student, and I then showed that patches are put into the DNA in wild-type cells, and then later was shown this does not happen if the cells are unable to take the damage out. The big step then, a few years later, was when Jim Cleaver in Bob Painter's laboratory in San Francisco showed that the disease, the sun-sensitive, cancer-prone disease, xeroderma pigmentosum, the victims of this disease were defective in repair replication. And this, of course, put the field of DNA repair in orbit. We could get our grants funded more easily. Actually, there's kind of a peak, and then it goes down again for many fields, but at least it was sure going great then. And as for the stability of DNA, Thomas Lindahl really peaked that area when, in 1972, he showed that guanines are lost from DNA spontaneously. Not that they just happen with RNA, but they just drop out of DNA. And in fact, each of you will lose 100 million guanines from your DNA just during my talk, and not because of it. So this was really important. Bob Painter also discovered unscheduled DNA synthesis in response to UV, and of course this turns out to be repair replication. And then a few years later, other excision repair pathways of base excision repair and mismatch excision repair were reported, and you'll hear from the next speaker more about base excision repair. You know, for life to even begin, there had to be certain requirements and required for persistence of the first living cells. There had to be redundancy. 2 identical sets of the entire genome, as in the 2 strands of DNA duplex, are needed to preserve the essential information for cellular survival through excision repair and recombinational mechanisms. Beyond that, there also have to be options to ensure complete duplication of the genome in the face of many obstacles if you're going to generate viable daughter cells. In this figure, which I'm not going to go into detail on, I thank Graciela Spivak for designing this. We try to show all of the multiple things that can happen to the DNA. It's a wonder that we can deal with all of this, but obviously we do or we wouldn't be here. There's many types of different damage, and I'm not going to talk about specifics except to say that there's a term called the DNA damage response that is often used mainly to talk about double-strand break repair and the activation of ATM and ATR to regulate responses, but there are other inducible responses, and I prefer to use the general term genomic stress response to accommodate all of these. And important kind of problem that is a lot of recent research is on what happens with arrested transcription and arrested replication, and what happens when a replication fork encounters arrested transcription. It's an exciting area for study. Nucleotide excision repair has two pathways. Global excision repair that deals with damage wherever it finds it, GTR, and then a pathway discovered in our laboratory in the 1980s called transcription-coupled repair, which targets the substances that block transcription. And it's a different mechanism of seeing the damage. Whether it's global repair looks for bulky adducts and detects weakness in the DNA, and ease of opening up the strands. Sometimes the damage makes the DNA tighter and is not seen by global repair, but it is seen by transcription-coupled repair because it's really measuring can the damage get through the narrow groove to get into the polymerase. And an important example from environmental concerns is Aristolocy acid. This is an environmental carcinogen and nephrotoxin of worldwide importance. It comes in, its seeds are in wheat fields from the Aristocola plants, and they're very similar to the seeds from the wheat. And so in some places in the world where they're not separated, they appear in the bread. Also, the compound from this is a traditional herbal health remedy. The relevant DNA adducts accumulate over many years in the silent genomic regions and non-template strands of expressed genes. They're only repaired in the transcribed strands of expressed genes, and so it's a very serious environmental mutagen carcinogen. Professor Arthur Grohlman, who is a pioneer and continuing leader in this field, was awarded the 2011 EMS Award for his work. The patients, this is xeroderma pigmentosum patients on the left. And these are obviously very sensitive to sunlight and it's extremely carcinogenic, 1,000-fold higher carcinogenicity from UV. They're defective in global genomic repair. The two on the right, Cockayne syndrome and UV-sensitive syndrome, are only deficient in transcription-coupled repair, and curiously, they're highly resistant to cancer. And we don't really understand details of this, but it's something to study. The patient on the far right only has sunburn and freckles, no cancers. There have been many exciting advances in basic science in recent years in this field. the highlight really is CRISPR-Cas9 genome editing, where you can knock out or knock in any place in the genome, make a point mutation anywhere. And in the past, we otherwise, of course, just generate as many mutants as we can and study them as we find them. But now you can make just the ones you want, precisely located. And people in laboratories tried to do this for years unsuccessfully, only to find that cells are able to do it all the time. So this is something to look for. If it's really important, biology have already evolved to do it. We don't have to figure it out for ourselves. But this allows for study of precise changes in these sites in enzymes and to look at the effects of changes in molecules in crystallographic detail, such that this can help with design of small molecule inhibitors. Another area that's coming up of importance are phase-separated aqueous cellular environments. It's not just membranes that divide the cell, but rapidly changing regions that can be separated as phase-separated environments. Also, unique gene expression patterns can be revealed in single cells. You can take one cell from a tumor and determine its transcriptome. And that information could be used to control growth of selected classes of cells, such as those in a tumor, without affecting adjacent normal cells and tissues. Basic research interfaces importantly with genetic toxicology, identification of chemicals and radiations that cause genomic instability and the relationships to human disease, determination of toxic exposure levels, risk assessment and exposed human populations with warnings and regulations on acceptable exposure levels. And the example that I'm most familiar with is Proposition 65 in California, which was enacted in 1986. Chemicals listed as known to the governor of the state of California to cause cancer and reproductive toxicity in humans or laboratory animals. The list has about 4,000 chemicals right now. It also has ultraviolet light, which means that you really should have a sign on the door, inside door of every building, because you go outside and are exposed to ultraviolet light. So there are some problems with the practice of how you use this list. It also doesn't tell you what amounts of these chemicals, how serious are they? I mean, do you have to drink gallons of the stuff in order to have an effect or what? And so the goal should be to provide a rational list with quantitative indications of genotoxic risk. But chemicals should be removed from the list if they turn out not to be genotoxic for humans. And a classic example is saccharine, an artificial sweetener that was shown to cause bladder cancer in male rats, not in female rats, not in mice, male or female, and there was no epidemiological evidence that it is any problem for humans. We now know from work of Sam Cohen and others that these crystals are formed in the bladder because the saccharine interacts with a protein that nucleates formation of these crystals. They irritate the walls of the bladder, and that causes the cancer in male rats. But it's no problem for humans. It took some years to get this delisted from this list, and this should be an ongoing practice to determine from mechanistic information what really needs to be on the list and what levels are important as a significant threat. Challenges for the future in the next 50 years, the continuing need for research on human genetic diseases with deficiencies in genes relevant to cancer development and aging, to define to our satisfaction and that of the public the dose-dependent toxicity of toxic substances such as those tabulated, which ones are the most serious threats and which are the ones we are best able to minimize. The EMGS outreach efforts should be greatly expanded and should include more engagement with the general public. Outreach is the key word, and is not merely a committee assignment for the Alexander Hollander Outreach Committee, but should be a responsibility for each member of our society. Thank you.